Welcome everybody to the Medicon Valley Alliance R&D Network webinar. We are soon starting, but just before starting, I'd like to give you a few of our household rules. So you're automatically muted. You can use the chat for technical questions, but please use the question and answer function for questions to our speakers. And you can write down your questions anytime, also during the talk, and we will pick them up so that in the Q&A session, in the end of the talk, we have some questions ready for our speakers. So there is no Q&A session totally in the end of the webinar, but right after every speaker, there is a chance for writing your questions down. You can also comment on questions from other participants, so that there is maybe some aspects or things that you think are particularly interesting or are linked to that question, then you can write them down there. And in the end, we will do a short poll with a few questions about the event, and that would be really lovely if you give us our feedback. I also would like to pay attention to the fact that we are recording this session, makes that we can publish it on our website afterwards, and you're very welcome to share the link afterwards in your networks. Thank you for that, and then we can start this webinar. Petter Hartman, our CEO at Medicon Valley Alliance, will say a word of welcome. Welcome, Petter.
Thank you so much, Sophie. And also for me, a warm welcome to all of you to MVA's R&D Network meeting. This is a network that we run at MVA together with our members, Alligator Bioscience and Medicon Village. And the idea with this network is to provide a forum where we can share our knowledge and to discuss current challenges for R&D and industry-academia collaborations, as well as aspects of commercialization. And our ambition is, of course, to stimulate discussions in a way so we can actually see some future collaborations coming out from the interactions between our members within this forum. For those of you who have participated in the R&D network before, you know that normally we used to meet in a physical setting, either on the Danish or on the Swedish side. But due to the current COVID-19 pandemic, as you know, it's not possible at this time to meet in person, and that's of course a pity. We hope that we can go back to normal with our meetings as soon as possible when things are looking a little bit better, and let's cross our fingers for that we can be able to go back after the summer. But for now, this is the way we will carry out our different network meetings, and we have actually in total 5 different networks at Medicon Valley Alliance that we run. And besides the R&D network, we also have an oncology network, a medtech network, a microbiome network, and a executive club network as well. And as a member of Medicon Valley Alliance, you are of course welcome to participate in these different networks free of charge. Focusing a little bit on today's meeting. Today's meeting is hosted by a research project called DiaUnion. It's a project that we at Medicon Valley Alliance facilitates, and we do it together with Lund University, Steno Diabetes Center, Region Skåne, and the capital region of Denmark. And I think this is a really good example of a strong cross-border research project that could really help strengthen the research capacity in the region in the future. And today's meeting will focus on connecting R&D of autoimmune diseases and therapies, and we will start with a focus on type 1 diabetes. And you will very soon hear more about the DiaUnion project. We have our first speaker will be Kristensen, who is the project manager for the DiaUnion project. And then we will have the opportunity to listen to Jannik Hilsted from the Novo Nordisk Foundation, followed by Åke Lernmark, professor at Lund University. And then we have 2 pitches, one from Adam Steensberg from Zealand Pharma, and one from Alenfall from Follicum. And finally, we will round up with a testimonial from Jeannette Söderberg from JDRF. And thank you to all the speakers for joining us today. And thank you to all of you who have signed in today to be part of this meeting. And I hope you will enjoy this session. And with that said, it's time for me to hand over the microphone and the screen to Finn Kristensen. Please go ahead.
Thank you, Peter. And thank you for all for joining. I will see if I can get my slide on right here. There we go. All right. So yes, I will talk about DiaUnion. I'll talk about the visions of the project, the goals, and the status of the project. But first of all, let me give you a little bit of background around type 1 diabetes. So type 1 diabetes is indeed an autoimmune disease. It is a disease that destroys, where the immune system of the body destroys the beta cells that produce the hormone insulin, which is necessary for nutrients like glucose to enter the cells. It affects randomly, often in the younger years, children and adolescents, and it is completely independent of personal lifestyle, unlike its sibling type 2 diabetes. It's a chronic disease which cannot be cured, and before 1921, where insulin was discovered, it was a death sentence for people who got the disease. Unfortunately, for many people around the world, it still is a death sentence. In many third world countries, it's never discovered. If you have type 1 diabetes, you just die without really anyone really knows why. Today we have insulin, and that's great, but unfortunately it is still a very complicated thing to control your disease, because unlike, for instance, vitamin C and other things you can take as nutrition, you have to administer it in very precise amounts. For instance, if I compare with the vitamin C, if you take a little too much, you'll just leave the rest with the urine. But with insulin, it has to be very precise. If you give a little too much, then you will, in effect, get too low blood sugar, which is very dangerous. You can get coma, and eventually you can actually risk to die from that. And if you give yourself too little, then your blood sugar will be elevated, and on the longer term, you will have an increased risk of complications, debilitating complications like blindness, amputations, and nerve diseases. So it's very important to keep that on target. And as you can see from the small table, it's also very impossible because it is very complicated. So we need to do better. We need to do more. But first, if we talk about the size of the problem in Sweden and Denmark, in our local area here, we are in an area of the world where we have the highest incidence and prevalence of type 1 diabetes. In Sweden, we have approximately 50,000 people, and in Denmark, about 30,000 people with type 1 diabetes. And this number is actually increasing at a rate that is almost as nearing the rate of type 2 diabetes. So it is something that we have to look into. Furthermore, it seems like the time of diagnosis is still getting younger and younger, so that really young children today, to a higher degree, get type 1 diabetes. And to underline how important it is to keep track of the treatment goals, we can see that of the 3 billion Danish kroner that the disease costs annually to the society, 1 billion of that, 1/3 of that, is for complications. And of course, you get the complications because you do not reach the goals. And also, to the patients, it is a really huge burden. You regularly have a low and high blood sugar, hypo and hyper, as it should have said hyperglycemia, and then you of course run into the risk of complications. Research for and development of better treatments for type 1 diabetes therefore is really important. And if we look into the underlying mechanisms, there are a lot of common mechanisms that are shared with other autoimmune diseases. For instance, thyroiditis and celiac disease, they belong to the same group of autoimmune diseases, and they share the same mechanisms. So it's really important to actually look into each other's diseases and see what we can learn from each other's research. This has not been done to a very large degree earlier, because the effects of the disease to the patient is very different, and therefore specialists, it has been different specialists that have treated the different autoimmune diseases. But this is something that we aim to do in the DiaUnion project to exploit that potential further. And at both sides of the Øresund, we actually have very strong competences within immunity, screening, genetics, and type 1 diabetes and other autoimmune diseases. We're trying to, with this project, we're trying more formally to gather these forces to see how far we can go together. And of course, our long-term vision is to create a center of excellence around autoimmune diseases centered around type 1 diabetes. So if we look more closely on the vision in headlines, what we want to do is, as we can see from this diagram, the following. First of all, when you want to cure type 1 diabetes or other autoimmune diseases, one of the most important things to look at is actually prevention. And in order to prevent the disease from coming to the patient, you have to find out who is actually in the risk for developing the disease, and who should you select to try to see if you could intervene and avoid the disease from happening. That's why screening is really important, and we have a focus on developing public screening programs in the region. Of course, we'll share research equipment and knowledge across the institutions that are involved, and based on that, we will, in our visions, we will look into clinical trials of drug candidates to prevent and perhaps also intervene with type 1 diabetes in already established patients. But in addition to that, we also want to secure that we have a flow of talent coming into the field, so that we always have an appropriate basis of researchers to work with this. And therefore, we look at education and career as well. We are working together with different institutions, of course, the universities, but also Danish Diabetes Academy, to set up research seminars, joint research seminars, summer schools, and the like. Finally, of course, very important to collaborate with the industry. And we want to, with industry, of course, we want to see if we could do common projects, industrial PhD postdoc project, and see where we can, if there are opportunities to use drug candidates also from industry. So that's in headlines the visions for DiaUnion. If you go a little bit further down and look at the goals for the period that we have right now, we are funded for a period of 2.5 years, from 2020 to June 2022. We want to strengthen clinical studies, and we do that mostly by introducing 2 new research projects, where one is taking mostly place, is based on Steensberg Diabetes Center in Copenhagen, and the other one at the Clinical Research Center in Malmö, belonging to Lund University. And again, as I talked about earlier, we are establishing education and career programs, and we already have talks and agreements to do so with the Danish Diabetes Academy. So that's the status on that, basically. Then we want to look into, this is something that we have not done yet, but we are starting up on that, to look into closer collaborations for treatment and prevention. That focuses a lot on public screening. And to do public screening, we have to show that we actually are able to do that with a meaningful purpose. And then we need to work with different authorities on both sides of the Sound to make it available to the public. Also, we want to develop collaborations with our research networks in this field, and we already have one potential partner on board to speak later today, namely the International Type 1 Diabetes Organization, JDRF. And finally, again, we are working to set up partnerships with industry, and in that way create public-private partnerships. And to round up, I'll just give a headline, in headlines, a little bit of update on the projects that we have going. You will get more details about that with the speakers following me. But basically, the project that we are, the overall goal of the two research projects that we are running in the current, under the current budget period of 2 years, are divided up in two projects, but the overall goal is to see if we can find new biomarkers for type 1 diabetes. Today we do have biomarkers, and we can, with a pretty high accuracy, predict with a person if a person is in the risk for developing type 1 diabetes. But it's very hard to say precisely when that will happen, and that's the rationale for actually looking into see if we can find more biomarkers, because of course it is important to know whether it's in 1 year or 10 years that this development will take place. So we're looking on lipid species to see whether they can more accurately predict this risk. And that's divided up in two research projects, one at Lund University and one at Steensberg Diabetes Center. Lund University has a long history and long and very thorough experience in screening through many international programs. And at Steensberg Diabetes Center, they have a similarly acknowledged knowledge about genetics and analysis of these kind of things. So that's why we've divided up in two projects actually. And
Finn, there's also a question in the Q&A, so we have soon time for questions.
Yes, yeah, just to the final thing I'll say is that we're planning for a feasibility study. As we talked about, screening is very important. So we're planning for a feasibility study for an inter-regional public screening program, where we aim to establish screening of 2,000 children on both sides of the Øresund to see whether we can, to demonstrate that it is actually possible to do this and to find the participants and actually to get the results out that we like. So with that, I'll just round off, and if anyone has any questions, please feel free.
Thank you, Finn. That's really inspiring. There is a short question in the Q&A, maybe not so much about the DiaUnion project, but about the incidence of diabetes 1 in developing countries where there is no treatment and where the prevalence is low. Do you have any data on that, or do you know if there is any data on that?
I think that there are very little data, for the same reason as many people are not diagnosed with type 1 diabetes. I do not have access to any data from developing countries. Perhaps, I don't know if there's any of the other speakers that could allude to that later on. I don't have any, but I know that that it is probably very limited.
Yeah. And a totally different question, but if I am a company and I would like to enter into the project in the industry collaboration, what should I do? What are you focusing on?
I think when it comes to the industry collaboration, there could be different opportunities. Of course, drug candidates are always interesting to test and discuss whether that could be an option, but also different kind of test materials could be of interest to include in a collaboration. And I would say in general, please feel free to reach out to us and let us discuss that opportunity.
Great. Okay, thank you very much, Finn. We will start with our next speaker, and that is Jannik Hilsted. So we start the video for Jannik. There you go. And put up the presentation. So you are ready to, let's see where you are. Not at all there. Show all windows. Okay. Anyways, this is fantastic as usual. What we do is we take this one. There you go, and we change to the next one. All right, let's try it again. There we go. Yes. Jannik, please go ahead.
Thank you very much. Thank you for the invitation to join this exciting meeting. I'm going to say a few words about the CNO Diabetes Centers and focus on the model of collaboration between the public healthcare system in Denmark and the Novo Nordisk Foundation. Next slide, please. First, a few words, summary words about the public healthcare system in Denmark. Denmark is a country with 5.7 million inhabitants. We have a healthcare system like the other Scandinavian countries, which is funded by public sources, that is tax. And we have a free and equal access to healthcare in Scandinavia, which is a unique situation for Scandinavia in comparison even with the other European countries, not to speak of US and so on. And then a few words about the foundation. The Novo Nordisk Foundation all began with insulin. As Finn just mentioned, we had a situation in the early 1920s, which was very optimistic in that a potential cure for diabetes was discovered in Toronto around 1920. And around 1920, August Krogh, a Danish physician, was awarded the Nobel Prize for something completely different, namely capillary physiology. And he was invited to go to New York to give lectures, and he was a very receptive person, and he discovered that something was going on in Toronto. So he and his wife, Marie, went to Toronto in 1922. And the short story is that August Krogh was given the permission by Banting and McLeod to produce and sell insulin in Scandinavia on the condition that the surplus of money gained by production of insulin would be given back to the patients and not to the shareholders. So that was the beginning of the Novo Nordisk Foundation. And very soon after, August Krogh came back to Copenhagen. It was quite evident that if you had to develop insulin treatment in a healthcare system like the Danish, it would be a very good idea if you had an institution in which you were able to test new insulins in type 1 diabetic patients. You have to remember that at the turn of the century, in the early 20s and 30s, healthcare systems were not at the stage that they are at the present time. Even measuring blood glucose 6 times a day would be something that was impossible in a public healthcare hospital. So therefore, August Krogh and Head-On decided to build a hospital by way of money gained from insulin production. And the hospital that was built was the Sino Memorial Hospital, which is in the far right in this picture. It was built in 1932. And in the years to follow, you can see on the slide that additional facilities were erected in the area, and it was quite evident that just like nurses has changed over the years, these girls are from 1935, then you had to realize that something had to happen with this compound, with this way of organizing diabetes treatment in Denmark. So what we were facing at the early 2010s were that the merger that was created between Sino Memorial Hospital and Hvidovre Hospital in 1991 was to some extent a success, yet you had to face the fact that you had a complex ownership. You had Novo Nordisk, the company, you had the foundation, and you had the capital region of Denmark. And you were moving from a manufacturer-driven institution to an independent healthcare provider. And that was not really happening in the setup that was created in 1932. You had to do something. You were also facing a decentralization of diabetes treatment, and you were facing a declining number of patients in Steno Diabetes Center as things were around 2010. So what was discussed and what was created was an idea of making a new Steno Diabetes Center, which will actually be geared towards the societal and clinical development that not only occurred at that time, but which was always also anticipating what would be going on in the future. The ambition was to create an internationally leading treatment and research environment to benefit everyone with diabetes in Denmark. And furthermore, to contribute to fewer new people with diabetes and longer survival with better quality of life for people with diabetes. That was the vision. So how would you do that? This is a very central slide because it describes in a few words the model that the Seno centers are built upon. You have two partners in the center construction. You have the public healthcare system that owns and operates the Seno Diabetes Centers. The ownership is the public healthcare system. And the foundation, not the company, but the foundation gives money for a number of purposes. Supplementary treatment, which is treatment not being standard treatment, which is given by the public healthcare system, but new ways of treatment that are not recognized yet as the treatment of the future, but which may become standard treatment in the long run. We give money for clinical research, education, health promotion, and new facilities, new buildings. We have agreements for 10 to 12 years, but we have no fixed end date for the collaboration, which means that we may have another 10-year period and another 10-year period and so on, depending on the state of affairs and the way that the development has been going on. Financially, the 5 points that I've mentioned amount to about $1 billion US dollars or 1 billion euro over a 10-year period for all 5 centers in Denmark. So it's a fairly large amount of money. The first center to be agreed upon with the capital region of Denmark was Diabetes Center Copenhagen. It was decided to have the center built, as you can see, in the immediate vicinity of a university hospital, which is Herlev Hospital in the outskirts of Copenhagen, so that the patients could have the advantage of being part of a university hospital, but still have their own identity, as the building sketch which you see here indicates. The next slide shows that when we came ashore with the agreement with the capital region of Denmark, it was decided by the board that we would go out to the other 4 regions in Denmark in order to ask whether they would be interested in having diabetes centers in each of the 4 regions. And fortunately, that was the case. So over the next few years, we had very intense collaboration and negotiations with the other 4 regions in Denmark, and we have succeeded in having agreements in all 5 regions now. And the way that these agreements are described is exactly the same as the one in the capital region of Denmark, with the same premises and the same amount of money given to each region depending on the size of the population. So finally, what are we going to do about the collaboration? We have a collaboration system within Denmark which is working fairly well between the Steno centers. No miracles have happened, but we're working on it, and I think that in the long run we will have collaboration. We also, of course, need international collaboration. We have had over the years a tradition for extensive collaboration between Danish diabetology and international partners. And the foundation is very keen on continuing this, facilitating research collaboration between the Steno centers and international partners. Exchange programs have already been established with international institutions, and we very much look forward to the option of having a discussion relating to collaboration across the Medicon Valley region. Thank you very much.
That was very interesting, Jannik. Thank you very much. And of course, there's also a few questions for you. And one of the first would be to hear if there is also a possibility to finance any research in other autoimmune diseases or the connection to diabetes.
I think the option is very, there is a very good chance, but of course you have to realize that you have to have clinicians that go into the project. You have to have diabetologists in the Steno centers that are willing to do the job, to devote the patients and the facilities in order to have projects running across different institutions. We have a Steno collaboration grant, an annual grant of $10 million US, which is given to collaboration projects across the centers. And in this context, other institutions may join as well. So the chance of having other Scandinavian institutions going into an application is absolutely there. And we also have open competition grants in which you can apply. And we are very much aware that the international collaboration is an indicator of whether we have a success or not.
Sounds indeed like water on the mill of Medicon Valley Alliance. Great. Thank you very much, Jannik, for your talk and answering the questions. We're going to the next speaker, and that will be Åke. Åke, you can connect. Indeed, welcome here, and you can share your presentation. We look forward to hearing you. There you go.
Thank you so much for inviting me to give a summary of present research on the origin or etiology of type 1 diabetes and celiac disease. And I'm speaking on behalf of the DiaUnion colleagues, Daniel Agard, Alexander Lind, he's a DiaUnion postdoctoral fellow, Fleming, who is a PI in Copenhagen, Sara Juel Mansax is the other, the Danish counterpart of the postdoctoral fellow, and Jesper Hansen Finn, you already heard, and Jesper is a physician at the Steno Center in Herlev. So Sophie, you have to tell me how to move the slides.
Yes, Åke, you can just use your arrows on your keyboard. If you with the arrow just go up and down, that should be feasible. Does that work?
Didn't work.
No. And what about if you take it out of presentation mode? Yeah. Can you change it now? Neither. Would you like me to put it up? That's also a possibility. Let me see if I can find your presentation, and then we can.
Now it works. Okay, I got it.
Great.
Sorry about that. So the humans are affected by a very large number of autoimmune diseases. It's almost like we know that almost every cell in the human body can in one way or another be attacked by the immune system of an individual. And there are over 100 different types of autoimmune disorders. And it's been estimated, especially in the United States, that about 16% of the population is affected by the immune system attacking our cells. And in the DiaUnion, we have formed a triad of autoimmune diseases, which take its beginning in type 1 diabetes. And the islets of Langerhans are, of course, in the pancreas. And that's why the GI tract is the target for this particular disease. Another GI tract disease is celiac disease. And that is an immune attack on the intestinal mucosa. And finally, the thyroid is well known. It was one of the first autoimmune diseases described, that in the thyroid gland, the immune system is infiltrating over time, eventually leading to deficiency in thyroid hormone production. So the goal of DiaUnion and our research is to explain the etiology of the disease, as well as the pathogenesis, leading up to possible ways of both preventing and treating the disease. So if you summarize the triad, type 1 diabetes is because the pancreatic beta cells are destroyed. Insulin therapy is the life-saving treatment. In celiac disease, the intestinal mucosa is destroyed, promoted by the presence of gluten. And gluten-free diet for life is the treatment. And about 10% of type 1 diabetes, of newly diagnosed type 1 diabetes patients, also develop celiac disease. In autoimmune thyroid disease, the thyroid is infiltrated by immune cells, and the production of thyroid hormone will cease. And thyroid hormone replacement, which is one of the most common replacement therapies in our society, is the treatment. And remarkably, more than 25% of patients with type 1 diabetes also have thyroid disease. And about 5 to 10% of patients with celiac disease also have autoimmune thyroid disease and require Levaxine treatment. So these are the examples of the cells of the immune system which are involved in the etiology of type 1 diabetes, leading up to the pathogenesis, which is the disease development phase of autoimmunity. And in type 1 diabetes, throughout the years, 4 different biomarker autoantibodies have been developed, standardized, validated, and they are autoantibodies to insulin, to GAD65, glutamic acid decarboxylase 65, GADA, and IA2, a protein in the beta cell that sits in the vesicle that stores insulin, and then the zinc T8 autoantibodies, which is a transporter protein inside the cell providing zinc for insulin crystallization, to make it simple. So these different cells with the dendritic cells in the center as the coordinator, presenting an antigen to helper T cells and the same or similar antigens to the cytotoxic T cells, interacting with the B lymphocytes that is stimulated by the CD4, the helper cell, to make autoantibodies. Those are the necessary components of the immune system to develop autoimmunity. And what we have is the biomarkers of this reaction, and they are the autoantibodies. Unfortunately, we don't have any reliable cellular measurements to find these cells or the CD8 positive T cells. But I'm sure those are methodological problems and timing problems that will be solved in the future. So autoantibodies are biomarkers of a just initiated pathogenesis. And the good news is that both EMA and FDA are in the process of certifying these autoantibodies as biomarkers, which will open up the possibilities to clinical trials in a completely different new way that we haven't had before. So in the insulin autoantibodies, GADA, IA2A, and zinc transporter antibodies are standardized, and they predict clinical onset of diabetes. And they are being used in prevention trials, not as endpoint, but to select participants in secondary prevention trial. Tissue transglutaminase antibodies are standardized. They predict celiac disease, and it's also a development primarily in the United States that these autoantibodies are used as a diagnostic criteria for celiac disease. Thyroid peroxidase and thyroglobulin autoantibodies are also standardized, widely used in the clinic, and they predict autoimmune thyroid disease. And it's all about timing to understand the etiology. It was known that these autoantibodies could be detected years before clinical diagnosis of either one of the 3 diseases. When then to start investigating etiology? And I would love to know the answer from the participants in the seminar, but for us, it was at birth. And back in beginning of the century, around 2000, the National Institutes of Health issued a request for application to start the study from the time of birth in children born with an increased genetic risk for developing type 1 diabetes. And as you will see, celiac disease and thyroid disease were tagged on as the study progressed. We screened 440,000 between 2004 and 2010 in 6 different clinics, 3 in the United States, Finland, Sweden, and Germany. We enrolled 8,676, and we followed them every 3 months for these autoantibodies. And the question we were asking, of course, what is it that makes an autoantibody to develop, and when do they develop? And we were surprised and taken a little bit by surprise, because the autoantibodies, especially to insulin, as you can see on the incidence rate of insulin autoantibodies first, that's the blue line, that they appeared very early. So already around 1 year of age, we had the peak of insulin autoantibody first. But by 5 years, 6 years, now 10 years of age, these autoantibodies are no longer appearing as the first autoantibody. On the other hand, we've also discovered that there were likely to be another endotype, and that was GAD antibodies first, and that's the red line. And you can see that the incidence of these autoantibodies are showing up later, and they continue. So even now in teddy children being 10 to 15 years of age, we have children who have been autoantibody negative for 10, 12 years, and suddenly they come to a visit and have developed GAD autoantibodies. So it's still out in the society an active disease. This is the timing of autoantibodies, comparing islet autoantibodies at the top with celiac disease autoantibodies at the bottom. And you can see that the autoantibodies come early. In diabetes, they stay on for a while, and this is the incidence of diabetes born out of these antibody-positive children. So you see the peak of developing antibodies are here, but the disease development continues and come later. So that the message is that you carry these biomarkers during a long time until the clinical diagnosis. Similar for CD, here is the antibodies, here is the disease. It's a slight delay, although celiac disease is much earlier. And the research is going on to find out what the pattern is for autoimmune thyroid disease. So what happened to the child prior to the appearance of the first autoantibody? So the most important part of the study we published in December last year, and that was the complete sequencing of monthly stool samples in 8,654 stool samples. All the virus was sequenced, and we had 383 children who had developed a first autoantibody, and to each one of these children there was a matched control who did not. And you can see we found 600 in the stool sample of these newly young children. They are 3, 6, 9, 12 months of age. We found 621 different virus taxa, and 72% of them were bacteriophages, and 20% of them were human mammalian viruses, and 8% were virus from the food stream, usually plant virus. And then we asked the question, when Yes.
Okay. There is also a little bit of time for questions, and that's coming like in 1 to 2 minutes. All
Yes,
right?
that's good. I'm done. So I just want to show you the first appearing antibody was IAA for enterovirus. So enterovirus infection was associated with later appearance of insulin autoantibodies, gastroenteritis like norovirus, or adenovirus F was associated with GAD as the first antibody. So GADA, the first biomarker, is preceded by adenovirus, and there are other changes in conjunction with this infection with biomarkers in the plasma that we have recently obtained information. And IAA first was enterovirus, and you can see there is a completely different pattern of metabolites and protein biomarkers. So there are two endotypes in type 1 diabetes, IAA first or GADA first. And tissue transglutaminase, similar data is being observed. Gluten and the amount of gluten is necessary, but it's not sufficient. A virus infection is needed. And we don't know anything about how these events may occur in thyroid disease. So the future direction is to find out what is in the black box between a virus-infected beta cell and the appearance of the first autoantibody. That is a key to the cure. Vaccination against the enterovirus B, Coxsackie, has already been initiated by Prevention Bio in the United States. Primary prevention trials are ongoing with oral insulin, probiotics, and gluten-free diet. And secondary prevention is the screening demo in the DiaUnion. So thank you. And these are the different sponsors of the TEDdy study and DiaUnion. I hope there is half a minute left for questions.
Fantastic, Åke. That's exactly what we needed. There is a question about how many viruses are there in healthy children compared to children with autoantibodies? I don't know if that makes sense as a question.
Yeah, so these are common viruses which tend to infect all children, and the highest frequency of virus are must adenovirus, which is about 18% of all the participating children, whether they had autoantibodies or not, were infected. So it is the difference between the controls when it comes to the enterovirus B for IAA first is that the children developing antibody took much longer to neutralize the virus than the control children. And the same for GADA first. Adenovirus F was being released to the stools in a prolonged fashion, and the children are not as efficient to generate or produce neutralizing virus to clear the virus infection as the controls.
All right, that sounds clear indeed. There is another question about, are you considering more initiatives in terms of diagnosis? Since often many patients with monogenic forms of diabetes are misdiagnosed as type 1 diabetes and therefore do not receive proper treatment.
That's a very good and important question. I think the numbers in Sweden, where we have almost 1,000 newly diagnosed diabetes patients younger than 18 years of age per year, it's almost 2% being monogenic diabetes. And there is a program already initiated in the 42 pediatric clinics in Sweden that every child is autoantibody tested and HLA typed. And those who are negative and do not have the increased HLA risk, they go on to be analyzed for monogenic diabetes. Very important.
Yeah, exactly. Åke, thank you very much for your contribution to this webinar. We will now get to the session which is called The State of Medicine Valley, with two short pitches from one company from Sweden, one company from Denmark. And we will start with the company from, who did we do first actually? Adam, please, Zealand, a company from Denmark. There you go. And you can start sharing your screen also. Fantastic.
Okay, do you see the screen?
Yes.
Okay, thank you very much for the opportunity to present what we are doing to change the life of patients and here at Zealand Pharma. And in this small pitch, I will focus on what we are doing to help patients living with type 1 diabetes. And really, I would say the focus that we are having right now as a company is, in this context, to bridge the gap until we have a cure for patients living with this disease. I've been with Zealand Pharma for just around 10 years, and I'm heading up research and development, and I'm also the Chief Medical Officer. This is my forward-looking statement, which we always have to present as a public company. We are a peptide-based research and development company who, just as of April this year, has also become a fully integrated biotech company with commercial operations in US. We acquired an established commercial entity in US, and thus we now have around 200 employees in Denmark and 150 employees in the US, most of which are commercial people. Our focus has been, over all these 20 years, to develop new peptide-based medicines that can change the life of people living with various diseases. And over the last, I would say, 10 years, the focus has been within metabolic and GI diseases. As I've said, we are focused entirely on peptides. So that is a skill that not many companies have been focused on for over the last many years. But of course, we are in the Danish and the Scandinavian region, we actually have a quite strong peptide-based drug discovery and development base. And for 5 years or so, we have had a very firm strategy of establishing ourselves also commercially, and thus building a strong late-stage pipeline. And that is, you can say, where we are right now, and I'll come to that on the next slide. So this is an overview of the pipeline that we have today. And as you will see, then we have 4 programs in the top, which are all focused around one molecule called dasiglucagon, and that's the molecule I will talk more about. The other franchise of our late-stage portfolio, that is around treatment opportunities for patients with short bowel syndrome. And here our long-acting GLP-2 analog glepaglutide is leading our efforts to help these patients. So that's a program that we have in phase 3 development, where we expect results next year. And then after that, we have an early program in phase 1 with a dual acting mechanism. Then we have partnerships with Boehringer Ingelheim with a molecule for treatment of obesity, type 2 diabetes, and NASH. And we also have a partnership with Alexion with our complement C3 inhibitor for various inflammatory diseases. And then a rich early pipeline, of which I've just shown a few examples below here. If we go back to dasiglucagon, then Jannik, he talked a lot about insulin and how Steno was founded based on the discovery of insulin, and patients with type 1 diabetes, of course, needing that because they've had an autoimmune attack, as Åke talked around, on the beta cells. But patients with type 1 diabetes, they also have you can say, and they cannot produce enough glucagon, the counter-regulatory hormone to insulin, if they take too much. So this relates very much to what Finn talked about in the start. Even though that we have a very good treatment for patients who lack insulin, it is extremely difficult for these patients to handle that disease on an on a daily basis, and you have to be extremely precise with the amounts of insulin you inject if you should not end up with low plasma glucose, with the risk of severe hypoglycemia, and etc., etc. Because if you just take a little bit too much insulin, then you have far too much, and the glucagon response that you can produce yourself is not sufficient. So dasiglucagon is a stable analog of glucagon. So glucagon has been available as a rescue treatment for many, many years if you have an insulin shock or severe hypoglycemia. But it is stable in nature as a molecule, and what we have succeeded with is making a stable version which can be in solution. And earlier this year, we actually submitted our first NDA to FDA, which is a rescue pen. So it's a little bit like an EpiPen to treat anaphylactic shock. So you just take off the cap and then you inject in an automated fashion. A patient with such a patient experience a severe hypoglycemic event. The same molecule is also being developed to be used as a chronic infusion, both for small children with congenital hyperinsulinism. They suffer from the opposite of patients with type 1 diabetes. They produce too much insulin and are therefore at the risk of hypoglycemia constantly, and we are then infusing dasiglucagon to counteract the effects. So that's the program in phase 3. I would say the most transformational opportunity we have in our pipeline is the dual hormone artificial pancreas that we are developing together with a Boston-based device company. So many of you probably know that there are insulin pumps available on the market today and glucose sensors, which have provided tremendous benefits for patients with type 1 diabetes, because it becomes easier to get to target the plasma glucose. It's still a huge struggle because you have to mitigate if you take too much insulin, or if you don't eat as much food as you had expected, or if you decide to walk instead of taking the bus. So what we are doing with this device company called Beta Bionics is that we have developed dasiglucagon as a stable solution in a small cartridge that can then sit into the second chamber of this pump. So this pump not only provides insulin, but it can also provide dasiglucagon pending, you can say, the glucose levels as measured by a sensor. And thereby, as we have shown in a phase 2 study, you can see the results here to the left. We can, in an almost entirely automated fashion, achieve very, very strong glucose control, getting, this was a short-term study, but if you kind of calculate the numbers of patients that could reach target, then we had around 90% who would reach a glucose target that is recommended by the American Diabetes Association, versus only 50% in the insulin-only group. And we achieved that while the patients had much less risk of hypoglycemia. And I would also say they had less burden because the device in an automated fashion took care of their lows, so instead of having to snack every time you are at risk of hypoglycemia. So this is a program which we really put a lot of efforts behind, and we truly believe could transform management of diabetes, perhaps not to the same extent as insulin did when it was introduced, but really still be something that we see as a major new innovation, hopefully coming to the market and to the patients in the coming years.
Thank you, Adam.
So with that, that was my presentation.
Great, thank you. And a very good presentation, putting the links also to the former presentations, and we see clearly the links. Thank you for that. Jen Alenfall from Follicum will be the next pitch. And there you go, Jen.
Yeah, so can you see the screen?
Yes, but it is you have to put it, into the presentation mode where we can only see your screen and not the preview.
Can you see it now? No. All right, I'll try to share it again. There we go.
Yes, perfect.
Okay, good. So thank you very much for inviting Follicum to this pitch. Follicum is a small company based in Lund, Sweden, and we are a public company also. So I have the same sort of disclaimer and as Adam had. So basically what we are doing, we're working with peptides also, and the company was founded in Lund 9 years ago. We have one clinical stage project. It's in phase 2 at the moment for alopecia, and the other one is diabetes, which is in early preclinical stage. And this project was actually started by serendipity. So we are working, we have identified interesting peptides from 2 different proteins coming from our cells. So the peptides we are working on are of endogenous origin, and we have found quite interesting results so far. So I'm doing this talk a little bit differently because I'm showing preclinical data in the following slide. So we identified a peptide that after subcutaneous administration will give a rise to quite a huge uptake in pancreas. And then in these are mice, and the picture is 1 hour after the administration. And after that, we analyzed where the peptide went into the pancreas, and we could demonstrate that the peptide was bound to insulin-producing cells in the Langerhans islets. As you can see in the picture to the right, the red is the peptide. It's very much focused to the Langerhans islets. It doesn't bind to something outside, so it's very specific uptake into the pancreas. We have also used a fluorescently labeled peptide with an in vivo imaging system. So this is a mouse where the peptide was given IV in the tail vein, and then we have followed the same mouse for times up to 48 hours. So we could here demonstrate that the peptide is localized in the central part of the mouse, liver, pancreatic area. And we have also taken out the organs, and we have also seen the same uptake in the pancreas after these experiments. So quite interesting that the peptide could actually be located so specific to the pancreas. So then we went on looking for the effects on INS1 cells. So a cell line from rats, a beta cell line. So this is a picture showing the appearance of the cells in culture. In normal glucose, you can see a nice sort of carpet with the cells. But in high level of glucose, you can see that many cells actually have disappeared, being lost, and we can see a lot of apoptosis and necrosis. So this is after 3-day culture in high glucose, and this picture shows you if we culture the same with the same glucose conditions after 3 days, but with the peptide, you can actually see that we restore the morphology of the cells and thereby protect the beta cells. The next experiment we did was to look for insulin secretion during the same conditions, that is 3 days in high glucose culture. And here you can see that the insulin release was normalized to high glucose control, and you see we have included a GLP-1, and you can see that, okay, the GLP-1 releases also somewhat larger levels of insulin compared to control, but some of our peptides did it even more. So we think that, you know, the peptides can protect the beta cells from the glucotoxic conditions, increasing the insulin secretion and also the insulin content into the cells. We also saw a decreased cell death during these conditions. So hopefully we can, you know, by administering the peptide, preserve beta cell, increasing their functionality, and maybe also restore the diabetic complications, leading to perhaps a better outcome of the patients. So in a next experiment we did together with Professor Lernmark was to use the BB rats. And you know, the BB rats are a good model for looking at type 1 diabetes. So the rat spontaneously gets diabetes around 55. And we did experiment with these rats. So we injected our compound 3 times a week from day 40. And we could demonstrate that the onset of type 1 diabetes was delayed by about a week. This is a very low concentration of the peptide. and potentially we could get even better results if we start the treatment early on. But this is what we have right now, and we are quite encouraged about these effects because it shows that the peptide could potentially also in vivo protect the beta cell destruction. And finally, in this experiment, we have also shown that the peptides induce a strong inhibition of the pro-inflammatory cytokines released from the immune cells of these mice, which could potentially be helpful in explaining the results of this, let's say, condition these rats suffer from. And finally, of course, we would like to see and understand how the peptides work in human material. So here we have donor material from healthy people, but also from type 2 diabetes patients. So here too, we have high glucose levels, looking at insulin secretion from our peptide, and we have compared with liraglutide. And we can show here that our peptide appears to secrete the same level or even slightly better than liraglutide on human Langerhans islands. And these experiments were also performed in collaboration with Lund University, but also a company in Switzerland. So altogether, we think that we have interesting effects of our peptides. And of course, as a small company, we would like to take this further. And that was my final slide. And thank you so much.
Thank you, Jan. That was perfect. Lovely for this short pitch from the Swedish side. Jeannette, it's your turn to give us a testimonial about JDRF and your activities. So you can start sharing your screen and turning on your camera for the last part. Yes.
I hope it's on, but I do warn you that I do have less bandwidth than I had hoped. So actually I'm going to turn it off, but at least you see me and you've got a face to the name and to the voice. So let me do this. Can you see my screen?
Mm-hmm.
Perfect.
But it's not in presentation mode, Jeannette?
It is in presentation mode, I believe. No?
Yeah.
Now?
Yeah, perfect. There you go.
Perfect. Well, so thank you so much for having me at this very interesting meeting. I've learned a lot this far, and I hope to give you some insights into what JDRF is and our thinking about partnerships and collaborations in the Scandinavian countries. So JDRF was funded in 1970 by two mothers who had children with diabetes. And in the 70s, we didn't know much about what type 1 diabetes was, and there wasn't a lot of research going into it. And especially there was no network of support for parents and families who had children with diabetes. So the two mothers, as you see here, together with NIH, started a foundation to raise funding towards type 1 diabetes research. And today, 50 years later, we are one of the world leading organizations in funding type 1 diabetes with a mission to improve lives today as well as tomorrow by accelerating life-changing breakthroughs through cure, prevention, and treatment of type 1 diabetes, as well as its complications. So I think that the important part here is improving lives today, as well as looking for a cure. We can't forget either one of them. Both of those aspects are extremely important. So we are the global leader in diabetes research funding. We've committed since the 70s $2.5 billion towards the research. We have around 75 clinical trials actively ongoing. And I think what sets us apart from other research organizations is that we have an in-house research team where we have 20 researchers, PhD trained scientists who are actually funding the research, but also giving strategic advice on where to go from here. You'll also see to the bottom left that we have attracted 2.8 billion in additional funding to diabetes research, and that's what we've done in the partnerships. And for the situation in Scandinavia, these are from last year, the numbers I've found. We're funding academic institutions and research groups. So your average basic and applied research scientist. But we will also invest in small and medium-sized companies to help the product to the market. We firmly believe that getting things to the market is the only way of helping the patients. We have one arm of JDRF, which is the T1D fund, which is a venture philanthropy arm, which does investments in companies in a bit different way of the foundation, where we give grants. They have an equity model that they're using. And in Scandinavia, or at least in Denmark and Sweden, because those are the numbers I took out, we've cumulatively funded 41 million in Sweden and 13 million in Denmark, plus the ones that we've seen to the bottom left. The 2 new grants were funded in Sweden of 6 million last year, and in Denmark an additional, bringing the total to 25 and 46. This is a space where we have a lot of people with type 1 diabetes. We have excellent researchers, and this is a space where we would like to see more collaboration and do more together. So as an organization, we always strive for partnerships and collaboration. We can't solve this problem alone, and we need to have trusted partners and friends to help us along the way. So the partners we usually collaborate with are pharma and medtech companies, but also other types of funding partners, VCs, etc. Associations, both patients and otherwise, but we also have links to the government and regulatory authorities, where we do have a policy work that we do in collaboration to try to advance the field of type 1 diabetes. The mechanisms of how we work together are very different. We're pretty much open to any idea that would advance science. We set up centers of excellence. We have targeted missions, for instance, the project of having an artificial pancreas out to the market. We run the clinical trials. We have, for instance, this is an American example of the special diabetes program, which we do together with NIH, where we fund $150 million per year for special populations with diabetes. And at the bottom, you see the healthcare pathway. And what we think is very important is that the research that we fund needs to come all the way through the pipeline to actually reach societal impact. Any kind of discovery research that only sits in a lab and never gets translated, approved, gone through coverage, and into the hands of a doctor who can prescribe it to a patient is a waste of time and money and excellence. The end user is the patient, and if you can't get the product to them, then we failed along the way. So we're trying to cover the whole pathway, and we also try to have a spread in the topics of our projects. So the ones that you see in here are the ones that we have together with the European Commission in the IMI setting. The reason I brought them up is that they are public-private partnerships together with multiple stakeholders, where we learn very much from each other, having different competencies and different backgrounds. It creates a melting pot with very much more to gain from the collaboration than if we had the same profile. You'll also see that we are covering basically anything that goes from immunology, cell biology, molecular pathways, through cell replacement, artificial pancreases. Especially prevention is a big part, and it links back to what Åke was saying before. Prevention is extremely important because the earlier we can get to the disease, the better. But we also look at improving lives of people who have the disease today. So everything that has to do with complications is a big importance to us, especially psychosocial aspects of complications, which sometimes have a tendency of being forgotten. And then on a systemic, like on a system level, you have the value-based healthcare, the health economics, trying to improve the burden of the disease, not only for the person itself, but as a society at whole. What can we gain from curing this disease? And while we do have this big spread of projects and we have the whole value chain and the whole pipeline that we're working through, I think we still have a laser focus on type 1, which is extremely important, that anything we do needs to advance science for type 1. That said, there are so many synergy effects to be had with other disease areas. Some of them are obvious, like we've been hearing today about diabetes and the autoimmune diseases. That's a given one. There are so many leads, so many good research projects that show that we can learn so much from the other autoimmune diseases. Some of these other collaborations are a bit more convoluted. If you look at antimicrobial resistance, you wouldn't think that there is an obvious link to diabetes. However, there is. The two main ones would be wound healing, which is an enormous problem for people with diabetes, with wounds that don't heal very well. And we have a lot of collaborations going on with people who are looking at that type of wound healing. The second part of collaborating with microbial resistance teams is that if they don't solve their microbial resistance problem, we can't go on with our immune therapies. We would put people with diabetes at risk if an infection would then kill them. So there are reasons to be in touch, collaborate, and discuss with other types of areas outside of only diabetes. And of course, then you have all the technologies, AI, the health economics, all of those that are overarching aspects that touch every type of research, not only ours. So where are we in Europe? Our strategy in Europe is to leverage the effect of having these partnerships, broad and deep. And we've identified a number of clusters in Europe where you have these regions of excellence, where groups are fairly close together geographically and can draw from each other and have these synergy effects. And one of the ones that we have identified as a fantastic area of opportunity is, of course, the Öresund region. And you are very well advanced, especially in screening in autoimmune interactions, which is an area of course, something of extreme importance. And I think
Jeanette, we're getting very close to the end of this webinar.
This is my last slide, so I'll leave you to it. The only point I'd like to make is that if you want to create a multi-stakeholder R&D cluster, you need not excellent academic scientists and the hospitals to be part of it, but you need the SMEs, you need the tech companies, you need all the stakeholders to go towards one goal. And I think that's the way we're going to move forward. So these, again, are the advantages. Of course, you know them, the accelerated ID generation and the bigger, bolder bets together. And looking to the future, innovation through collaboration and creating value through co-creation together with patient and patient organizations such as JDRF, we think is the way forward. So I'll leave with you that, and thanks a lot.
Thank you very much, Jeanette. That was a great testimonial. So we're at the end of this webinar. I would like to say thank you to all of you. And we launched a short poll asking for your feedback on this event. Thank you to all our speakers, Finn, Jannik, Åke, Adam, Jen, and Jeanette. Thanks for a great contribution, and we hope to see you soon again. I'll leave the webinar a little bit open so everybody can finish the poll in the time that they need. But I wish you a very good day, and see you soon again. Thanks.